So now finally we get into describing the structure of a community. This lesson is going to be the first of two parts, and today we're going to cover diversity indices. Next time we'll be talking about abundance in more detail. Recall from the first lesson from this course that the most fundamental attributes of a community are diversity, which is comprised of richness, which is the number of species present, and evenness, which is the relative abundance of each species. In other words, the number of individuals per species. We need to be able to quantify each of these in order to be able to evaluate management activities, to examine disturbance, and to otherwise monitor and compare communities. There are many, many ways of doing so with different methods or formulae that emphasize different aspects of a community. For example, some emphasize rare species. They want to make sure that they capture rare species. And other methods instead or sort of the mind of we need to make sure that the things that are common, that comprise most of the individuals of the community, that they are emphasized. So you have differences in that way. You also have different methods that are appropriate for different data forms. So if you have presence-absence data versus if you have actual counts data, if you have lots of zeros in your data or not, and so forth. And so we're going to go through all three of these components, diversity, richness, and evenness today. These are two books that I highly recommend if you want more detail about this topic. Both are by Anne McGurran, and the ecological diversity and its measurement is an older book. Measuring biological diversity is more recent, but both of them are incredibly detailed, and she does a great job of going through the rationale behind different methods, comparing them, going through the math behind them, and so forth. So I highly recommend these books. So let's start with richness, the most fundamental property of communities. Remember that communities are multi-species assemblages, and richness is the number of species present. It's usually abbreviated as capital S. Now, seldom do we know the true value of S in a community because typically we have samples. And from those samples, we can estimate what we think is the value of richness for a community. We can do that if we have counts of a species, of a number of individuals of a species at a single site. And those are called abundance-based methods because you're using abundance data. Or you can use the frequency of species occurrences across many sites. And these are more commonly known as incidence-based methods. Last time I discussed how some community patterns were based on occurrence, whereas others were based on abundance. Richness experience is the same thing. There are occurrence-based measures, called incidence-based measures, and abundance-based measures. Abundance-based measures are especially useful if you have actual counts of the number of individuals belonging to each species, as would be the case for these birds at this urban wetland. You can see the different species and you can count the number of individuals belonging to each. Incidence-based methods are particularly useful if you can't do that, like for example if you only have presence-absence data, or if you cannot otherwise make accurate counts of numbers of individuals, as might be the case if you have like clonal species like these plants and this meadow here. It's like how many grass individuals are there? Or for species where it's difficult to keep track of numbers of individuals, like if they have massive swarms, like for example this murmuration of starlings. These are these wonderful swirling synchronized flocks here. I tried to embed a video in this and it didn't work. So you can go to YouTube and Google murmuration of starlings and you'll see these wonderful swirling flocks, almost like smoke. And you can see how it would be very difficult to keep track of the number of individuals, and you would be in grave danger of either double counting or missing individuals. And in those cases, abundance-based measures would be full of errors, and so it would be better off for you to use incidence-based measures. There are many, many choices for both of these. These are some of the most common ones. These are the ones that I went through in the online notes for today. But most R packages give you an array of these, and you have to understand the limitations of each of these. So, for example, the Chow indices are especially useful if you have lots of singletons or doubletons in your data. Singletons are individuals, are species that are represented by one individual, and doubletons are species that are represented by only two individuals. However, both of these measures, the abundance and the incidence-based Chow metrics, tend to underestimate the true richness present in a community. The jackknife measures can overestimate or underestimate richness. Most of these richness indices will give you a value greater than the number of species that you observed, because the assumption is that the number of samples of species that you observed in your samples is lower than the true number. But jackknife can actually yield values that are less than the richness that you observed. And obviously, those are wrong. This is especially problematic if you have lots of zeros in your data. The bootstrapping method has a similar limitation if you have lots of zeros, if you have a sparse matrix, although it is less prone to overestimate richness. So in the notes I mentioned, each one of these has its own uses and its own limitations. And in McGurran's 2004 book, her Measuring Biological Diversity book, she summarizes the performance of these and some other richness indices. And there was no consensus as to which one was best. And so, therefore, what you need to do is consider the structure of your data. Is it abundance-based or incidence-based? Do you have lots of zeros in it? What's your sample size? And then pick your measures accordingly. And it would be prudent for you to use several metrics and then use the consensus outcome. So, for example, these plots are for the 5 columns of output for the spec pool function from vegan. In other words, your recorded S, the Chow2, Jackknife 1 and 2, and Bootstrap with 95% confidence intervals that are based on sample size. In other words, the number of sites that are sampled, because these are incidence-based. And so from these plots, you can see which indices behave weirdly, like looking at you, Chow2, right here. Look at this nonsense. And which ones consistently overestimate or underestimate richness. And here is what you observed. And so what you're doing is you're kind of comparing how all of these others behave in reference to your observed richness. If you have abundance-based data, once again, plot the multiple metrics that are available to you and just compare them. And again, we have the measure and the 95% confidence intervals around it. Notice how for both of these, they have wider confidence intervals at lower sample sizes than they do at higher sample sizes. That's true for both of them here. And that would be expected, meaning that estimates of richness from low numbers of samples may be especially inaccurate. So now that we've estimated richness, let's move on to diversity. And here's an example of why using a diversity index or a diversity metric, those are synonymous. Here's an example of why this is important. We have two communities here with species that I've represented in different little colored shapes here. And these contain the same number of species and the same number of individuals. But you can see that the two communities are not the same. And so therefore we need something beyond just S, richness, and N, the number of individuals, to denote this difference. We need a diversity index. Assays of just richness or total abundance would not accurately portray the differences in these two communities. Diversity indices can allow for such distinctions to be detected. The main problem with diversity indices is that they take richness and evenness, which may be under very different ecological pressures and evolutionary forms of selection, and they combine them into a single number. So the simplicity of a diversity index is simultaneously its strength as well as its main weakness. In the online notes today, I talk about many different types of diversity indices. Perhaps the most common one that's used is the Shannon-Weiner index, H prime. It's one of the most commonly used indices, and yet it is highly sensitive to sample size, it's highly influenced by rarity, and it can be difficult to interpret. So there's also the Simpson's index, lambda, and because it has a contrary behavior in that low values indicate high diversity, it's usually manipulated into different forms like the inverse Simpson or into the D index. And this D index is what Biodiversity R and vegan mean when they say Simpson's diversity index. This index is often recommended over the Shannon-Weiner index because it is less sensitive to rarity and to sample size, but it also can be difficult to interpret. Fisher's alpha is not sensitive to sample size if your sample size is over 1,000. It also assumes that the data follow a log normal distribution, which isn't always the case, and it is an iterative algorithm, and so I don't really cover it in any detail whatsoever because it is something that has more problems than, frankly, it's worth. And there are lots and lots of others. And every metric has its utility and its problems. So what you do is you calculate the value of each one of these for a given community. So if we go back to our little example of the two communities with the shapes, here are the values of richness, of the number of individuals, of the Shannon-Weiner index, and of the D, the 1 minus Simpson's index here. And you can see the differences between these two. Now, when I mentioned that one of the problems with these metrics is that they're difficult to interpret, what I mean is this. 1.22, there's no units to this, and D equals 0.67, there's no units to this. Is 1.22 really different from 1.61? Is 0.67 really different from 0.89? That's what I mean by difficult to interpret. And that's also, again, why using many metrics in order to find out what the consensus trends are in terms of which one is consistently more diverse than the other, why that is warranted. In our little cartoon example here, it is absolutely evident to see that this community on the left is dominated by the red circle species, whereas over here the community on the right is absolutely equitably distributed, the number of individuals across every species. We can see that in these communities, but we seldom can see that in our real data. And so the idea is you calculate many of these and you see, okay, they've got the same number of species, they've got the same number of individuals, but this one over here, this right community, has a higher value here, so it's going to be more diverse. And hey, and by Simpson's, it's got a higher value here than this one over here on the left, and so it's more diverse. And so therefore, I think that the community on the right is going to be more diverse. And by looking at this cartoon, we know that to be true. If you have replicates, as we did in the case of the beetle data that we worked with in the online lesson, if you have replicates, then you can show average diversity, and you can create a box plot as we did here. And from this you can see that grass was the most diverse habitat type according to the Shannon's index anyway. You may have noticed that in the online notes I will often have questions that are in blue text, bold blue text, and those are ones that are not part of your assignment. They're ones for you to sort of work through that I will then try to cover in these kinds of videos. And so, for example, one of those questions asked which of the habitat types was most diverse, and it's clear that it is grass. And so now let's turn our attention to evenness. As we noted earlier, these two communities have the same richness and the same total number of individuals, and yet they differ greatly in their evenness. And just like for richness and for diversity, there are multiple evenness indices as well. I don't cover very many of them, in part because P-lose J, which is also called the Shannon Evenness Index, is so widely used. But our packages will give you an option of many others. Because the P-lose J is based on the Shannon Index, it is subject to the same kinds of limitations as the Shannon-Wiener Index. It's highly sensitive to sample size, and it's also highly sensitive to rare taxa. Again, there is no metric that is perfect. Instead, use a bunch, find the consensus. And so let's go back to our familiar example of these two communities. And here we have the diversity metrics that we had before, and now we have the values of P-lose J. And you can see here that the one on the right has a value of 1. That is perfect evenness, which you can see in these data. So with these two communities, and with the values that we calculated for them, you can probably detect that there are going to be problems with these, and there are some well-known problems with richness indices, diversity indices, and evenness indices. Perhaps the most well-known of these, the most problematic of these, is the fact that these are numbers here, and they do not take into account the identity of the species in question. So that's important if you're dealing with invasive species or exotic species. If you only pay attention to numbers, then that means that quantitative diversity measures may not differ between communities, and yet the communities themselves may differ profoundly in the ecosystem services that they provide if you have an invasive species or an exotic species that replaces a native species. Similarly, they don't distinguish between established species and vagrant species or transient or accidental species or one-hit wonders kinds of things. Things that are established that are consistently found versus things that are accidental. You see them once and you never see them again. So these indices don't take that into account. And then, as I mentioned before, how different is different enough? Is 1.22 really that different from 1.61? Well, without knowing what the actual scale of the data are, you don't really have any way of distinguishing that. If you do have replicate samples, as we did like in the ground beetle data, then you can calculate and you can see what the average is. And so again, it is beneficial if you have multiples and you can take an average, or if you can do multiple metrics and then take sort of not a mathematical average, but sort of an average consensus of what's going on. Okay, so what are you supposed to do? So because different diversity indices weigh species abundances differently, in other words, some treat rare species more, they emphasize them more than common species, and then other indices have the reverse problem. Or they have different data assumptions. The conclusion of whether a community is more diverse than another can depend on the diversity measure that was used. And so what you need to do is, first off, examine your objectives. Are you interested in looking at just richness or just evenness or everything? You need to examine your data. Do you have incidence data, occurrence data? Do you have abundance data? Do you have lots of zeros? All that kind of stuff. And then finally, you should use multiple metrics, multiple appropriate indices given your objectives in your data, and then look for consensus. If you get output from a particular metric that seems contrary to the consensus, then you should investigate that more. It's possible that your data violated the assumptions for that metric. And if your output doesn't have consistent trends, then that too warrants further investigation. I've been talking about diversity, and yet there are different scales of diversity, from local to biogeographic, that are due to different processes. So, for example, local diversity is the result of organism responses to local conditions. And then biogeographic diversity is the result of speciation. And so we really need to distinguish what scale of diversity we're interested in and what we're talking about. And so these are the different scales of diversity, alpha, beta, gamma, and epsilon, where alpha is the number of species within a site or a habitat, and it's quantified by the indices that we just talked about. Beta diversity is sometimes called turnover diversity. It's the difference in diversity between habitats or between patches, and it represents the extent to which species composition varies by location or by habitat. It is influenced by scale. It decreases as the area that you sample increases, either in space or time. And relatively higher values of beta diversity, indicate fewer shared species there are. In other words, you have low community similarity, whereas a value of 0 beta diversity would indicate total overlap across habitats, no turnover of species whatsoever. And it is quantified by specific beta diversity indices or similarity indices. Gamma diversity is sometimes called landscape diversity, or the diversity of species found within a region of multiple habitat types. And then finally, epsilon diversity is the diversity of a biogeographic province or biome. And so here's a cartoon to exemplify this. Let's say you've got 3 different patches or 3 different habitats, and each of those you can calculate diversity within them. The richness, the evenness, the diversity metrics, the evenness, all the stuff we just did. You can do that for each one of these habitat patches. That's alpha diversity. Then you can also compare the differences in these, and that is beta diversity. What species are shared among these patches versus which ones are unique to each one. And then you have the overall diversity, which is gamma diversity, the landscape diversity here, with a landscape being comprised of multiple habitat types. And then finally you've got the whole biome that you're talking about, and the biomes species pool, and that's epsilon diversity. So in the exercises for today, we've started by working with presence-absence data, the gbbiol.csv file of the ground beetles. And in calculating richness, we leaned pretty heavily on the package vegan throughout today's lesson. And in vegan, we specifically used a couple of commands of spec pool and pool accum if you have incidence data, or estimate R and esta accum if you have abundance data. For diversity, again, we used vegan, and there's the diversity command that you can use. But I also show some examples using the package plier, and then the package biodiversity R has its own commands like diversity comp. Now in the notes I mentioned how if you ran diversity comp you would get an error, and that you had to use a command called strings as factors. You had to take those text data, the strings, and you had to tell R, no, those are things that we can compare by. And so that's what the strings as factor command got for you. When you do your R markdown for your homework, R markdown will fail at any error like this. So make sure to remove any error or set aside such lines with the pound sign, the little hashtag sign. Otherwise, your R is not going to work. Okay? So, with your strings as factors, it works. And then there's also diversity result command with biodiversity R. And similarly with evenness, we used biodiversity R, and we used the same kinds of commands in order to get various evenness indices, including the chow, the jackknife, the bootstrap, ace and ice, the J index, and so forth. For scales of diversity, because beta diversity compares diversity across habitat types, not only did we use the GB BIOL data, we also used the GB site CSV file. And again, we used vegan spec pool, but also the beta diver command, which gives you 24 similarity indices, or actually dissimilarity indices, because they're looking at turnover or differences. And in the notes, I discuss how several of these are especially commonly used, such as Whitaker's W, Jacquard, Sorensen's, and then similarity is also recommended. Then, in order to determine whether there are statistical differences in this turnover, you can use a function called ANOSIM, or analysis of similarity. It's like analysis of variance, but in this case you're using similarity indices. There are several alternatives to ANOSIM that have different assumptions for different types of dissimilarity matrices. And these include various forms of permanova, for example. So on the one hand, ANOSIM does not assume equal group variances. However, it only allows for simple one-variable models with no interaction effects, and it can only be used for categorical, not continuous variables. And so ANOSIM has a lot of limitations. I'm just including it at this stage as an example of how you can statistically compare values of beta diversity. But given the structure of your data, you might look into some alternatives to it. And so this is how you do your ANOSIM. In this case, I used the Whitaker measure of dissimilarity, as you can see. And then in order to see the actual output of this activity, I have to actually call this R object that I created, gb.ano, like this. And then the output that I get gives me this, and you can see the significance. It's highly significant here. What this tells you, this p-value, just gives you an idea that there's a difference somewhere. And so what I did was I also plotted this relationship here using plot like this, and this is the result that you get. And this graph shows that the dissimilarity between groups was higher than the differences within groups. So for your assignment today, you're going to make use of two new CSV files. One is butterfly presence absence. It's a site by species matrix. And the other is butterfly sites, and it's a site by environment data frame. These data are, the species are real, the sites are real, but the actual data themselves I made up. The butterfly species in the actual data set are real butterflies that occur in the northeastern United States, and the three habitat types, the bogs, fens, and meadows, also occur in that area. But which species is located in which habitat and which numbers, that's made up. So don't read too much into it biologically. But what you're going to do is you're going to take these data and you're going to do the most fundamental type of community analysis on them. You're going to examine diversity using richness, diversity, and evenness measures across different habitat types. And you're going to do a quantitative comparison of these three communities, bog, fen, and meadow. And in doing so, you're going to answer these questions. And these are absolutely fundamental kinds of questions that should be asked about any ecological community. How many species do you have? How many were in each habitat type? Which habitat type had the most? Then which richness indicator do you think gives the best estimate of what the true richness is, given that we have samples of that? Which habitat type is the most diverse and which was least? And provide some support for that. In other words, make a graph that would sort of help prove your point. Which habitat type had individuals that were the most evenly distributed versus which ones were more dominated? What was the landscape scaled diversity of butterflies? Which habitat type had the highest alpha diversity? And then which sites were the most similar in species composition? And again, be thorough and provide some kind of statistical support for your answers. And then finally, look for outliers. Were there any sites that were outliers with respect to butterfly richness? And if there are, you might consider removing them and rerunning your analyses and seeing how your answers change. These are the kinds of questions that are asked about any ecological community. And so now you have the skills in being able to answer these kinds of questions in your own data.